Welcome to the COVID-19 Researcher Spotlight Series. Today I'm joined by NEB Associate Director of Applications and Product Development, Eileen DiMolanta, and her longtime collaborator, Dr. Chris Mason of Weill Cornell Medicine in New York City. Chris really hit the ground running when the coronavirus pandemic began its global spread, and he started with metagenomics studies in Grand Central and Times Square subway stations, but he didn't stop there. He's now receiving public transit samples from around the globe. Chris has also been instrumental in bringing loop-mediated isothermal amplification, or LAMP, based SARS-CoV-2 detection methods to communities. And he's giving new meaning to the word GLAMP.
Chris, thanks so much for joining Eileen and I today.
Oh, thanks for having me. It's a pleasure to be here.
Thank you. I was hoping that you could tell our listeners a bit about your COVID-related research.
Yeah, happy to. So we've been happy as well as exhausted to tell you about it. So I'd say it's been a really crazy, few months ever since late February when it was really clear that the wave was coming our way in New York City. We got hit, as most people know, particularly hard, and it really started with just ramping up RT-PCR and methods for diagnostics, basic diagnostics at our pathology core, and then also at the hospital. The big ramp up in the early days was to get basic PCR methods up and running. But actually, in discussions with NEB scientists like Eileen and others, Nathan Tanner, actually, in the early discussions, even in February, like, well, are there other ways we could do fast diagnostics and testing? And some scientists from NEB and also researchers in China had a preprint out using LAMP, which is a loop-mediated isothermal amplification method that had showed that it worked on, it was only the first 7 patients tested on, but it showed that it was a possible way to detect SARS-CoV-2 in patient samples and looked really intriguing. So that launched a whole flurry of activity for the past 3 months on thinking about LAMP for hospitals, looking at environmental sampling in subways, looking in patient samples, partnering with other academic groups, industry groups, nonprofits, all getting a variety of methods out for rapid testing, which is LAMP, which we'll probably talk about a little bit more, but then also a more in-depth profiling of patient samples. So for every one of the first patients that came through the hospital, the first 735, we did for almost all of them RT-PCR and then LAMP and then also RNA sequencing with the NEB Next kits for ribodepletion and look at the total transcriptome to see everything from the NP swabs, the nasopharyngeal swabs, what's happening at the site of infection, and what can we learn about the virus, the host response, and also the RNA biology, really what's happening where the infection is occurring. So all that's happened, almost all of it with NEB reagents, actually. So it's all been really, really been powered by a lot of the supplies for rapid testing as well as in-depth transcriptome profiling.
Well, that's great. So how do those different methods compare? How does the LAMP method compare to the qPCR?
Well, this is one of our first questions. Is LAMP LAMP was first published by a Japanese group in 2000. So in some ways, it's decades old. It's not as old as say PCR methods in terms of development, but it's an amplification method that is a cousin of PCR in concept, except that as the name implies, it's isothermal, so you don't have to cycle through different temperatures. You can do it at the same temperature. There's even a Twitter post of someone who did it in his kitchen last week who if you heat a certain 7 deciliters of water to boiling and you add 3 deciliters of cold water, then you can actually run it on your stovetop. So it's very portable and amenable to low resource settings. And so, but because it is in some sense simpler, there's been a question of, well, how would it compare? So we did comparisons to RNA sequencing and RT-PCR and found a correlation of around 0.8 or 0.9 depending on which subsets we looked at. So it was very correlated in terms of a high viral abundance or medium or low. You could see pretty good correlation. And we also observed that obviously it's faster. The difference was that you'd have to look at very low viral abundance samples. We'd start to see more variation, but that's true for any method. So if you look at RNA-seq or LAMP or other diagnostic approaches, when you get low copies of any molecule, you get more noise as you get towards that sensitivity threshold. So, but we put this online, worked again with multiple NEB scientists, and some are some are on this call. So we put it online. It actually eventually worked with Color Genomics. They also took early versions of the protocol and got it working at scale on Hamilton robots, and it's now being used. It got FDA EUA approval, so now it's being used for diagnosing actually over 10,000 samples per day in San Francisco. So all that
Oh,
was
that's
like,
incredible. I didn't know they
yeah,
were up to that scale now.
yeah, yeah. Well, allegedly by text up to 14,000 was the maximum per day, but that was actually why we really wanted to start to work with other groups and researchers, academic sites, commercial sites. As And we've even started a consortium of LAMP researchers around, if anyone interested, because I kept my inbox was full of questions about our preprint and ideas, and so I said, how about we just have a weekly call with everybody, because I keep having lots of emails with the same questions. And so now we have a global LAMP consortium, or GLAMP, which is like a glamour camping. It's kind of like that, but nothing like that. It's very, it's just the name. But it's been an extraordinary time, because everyone, well, almost everyone, not everyone, but almost everyone is sharing protocols, methods, techniques, ideas, their failures, which are very important in this stage of a pandemic. Like, okay, what worked, what didn't work. Even on the call just last week, we've ways to do enrichments. We all know that SDS and other surfactants are bad for LAMP reaction, but someone was using glycol enrichment last week, which it was a, kind of an aha moment on the call. I was like, well, never thought to even try glycol for basically nucleating the reaction and increasing the stoichiometric density, so the reaction goes more efficiently. Which is like, oh, it's a good idea. So it's been a really lots of long hours, but lots of fun collaborations.
Chris, you mentioned your preprint and collaborations, and we've had calls in the past few months, as you said, and I think the pandemic's really forced uh, global collaborations. Can you speak a bit about what you think the value of these preprint services are, especially during the pandemic, and how do you see them being used like long term in the future?
Yeah, it's been really interesting on Twitter, which is a major platform for presidential policy announcements, as well as a great place for science exchange. It has been really extraordinary to watch the speed with which science has moved. It is, it's an essential platform. As I think most pronounced in evidence of it is that Nature, when we submitted the paper to Nature, you have to submit it to a preprint server when you submit to Nature. I don't know how many other journals have had this policy, but one of the flagship journals of all science is requiring it now. So it's interesting that I had an email exchange with, I will just call it an old school professor, in that he's over 60. I don't know. I'm getting close to old school. I'm like mid school. It's anyway, and he was actually insisting by email that he didn't want to put it on a preprint server yet, because he had this really big repository of NP swabs and other clinical metadata. And I said, well, actually, Nature requires you to. And he kind of couldn't believe it. So it's really, I think, for anyone that's been thinking about science in the past, 2 decades or even century, that idea is new, at least in the biological sciences. But keep in mind, for physics, they've been using Archive, the preprint server, actually since the late 80s. Like, it's interesting. It really depends on the field you're in. The biological and medical science is a little bit more new to this idea, but in the physical sciences, they've been doing it for a long time. So, but in the case of a pandemic, it's both good and bad, because really sensational reports can come out and then people kind of get distracted. But the better thing is, within a matter of days, usually there's enough comments. It's pretty much clear the field has commented on something, whereas it's actually worse and slower for most journals, like the chloroquine, hydroxychloroquine trials that this random group called Surgisphere, they published a paper in the Lancet, and then didn't release any of their data, any of their methods. It flew through peer review. It didn't really get peer reviewed because no one could look at the data. So it is actually, if anything, it's further highlighted the challenges of the current publishing system. That it's not. I mean, no system is perfect, but in the case of preprints, the corrections are a matter of days. Where with papers, it can take weeks, sometimes, or months, or even years. And so I think it's been great. It's been essential. Mostly
So
great.
you mentioned that a lot of your work was on nasopharyngeal swabs. Have you tested other sample types as well?
Yes. So since then, and even in the preprint that we put online, there was some oral pharyngeal. There is an ongoing debate about what is the best place of the body to trace the virus, and even our
Mhm.
fundamental knowledge of the tropism of the virus, or where is the density of the virus in the different parts of the body. The tropism just refers to the tissue specificity of where the virus goes and populates. We're still
Okay.
learning about that, about SARS-CoV-2. But we know, at least from as a group at Ann Wiley at Yale published a paper saying that saliva is better than NP swabs. Other groups have said that NP swabs are better than any other collection site, and they might both be right, because it might just depend on your state of infection and where you are on the trajectory of your infection. And so basically, the earlier time points have a lot more virus for almost all parts of the body. But the virus has been seen in stool as well. So we've begun looking at stool samples, including sewage. And most of the virus does not seem to be infectious, but you can see it in people even weeks after they have no symptoms. So, we've seen in other parts of the body, but we're focusing almost exclusively on saliva. Actually, some of the slogan for our working group now is "Saliva is the future." That's Ted Davis who has said that, but with that, we're hoping it's just so much easier to collect. So we know it can be useful and is either as good or maybe slightly worse than NP swabs. And if you think about testing, people going back to college, elementary school kids, large employee populations from whole companies and municipalities, like, you can't jam a swab up people's nose. It's risk to the healthcare worker. It's uncomfortable. It's the equivalent of a cranial colonoscopy with no sedative, is what I like to call it, which is totally gross, but I think illustrative of the process.
Colorful.
Yeah.
Yeah. It does sound illustrative of the process. Yeah. I think everybody would really appreciate having to do that less in terms of testing.
For
So
sure.
you also tested samples from subways to look at the metagenomics data. How did you collect those samples?
Those were at the Grand Central and Times Square, and that was being done right at the beginning of the outbreak in early March. And I have a penchant for subway sampling ever since 2013, when my daughter would put things in her mouth that she grabbed on the subway or lick subway poles. I became immensely curious. One of the best parts about science is just to be very curious about things, because it leads you to interesting places. So that's been part of an ongoing study to look at the genomics and also RNA viruses that are on different surfaces around the world in public transit spaces. So there, we so far tested negative. There was actually one lamp reaction that turned slightly orange, but it didn't show up when we sequenced it. So we think it was really, even by lamp or RNA sequencing, we don't really see much evidence of the virus in the early days. However, we did just finish a big hospital study with Northwell and looking at patient rooms before and after they check out that are COVID positive. And we're using LAMP right there to test it in the room, and then also doing sequencing and RT-PCR. And we can for sure see some of it in the hospitals. It's not surprising. There are patients there. But the other thing we're kind of piloting out is getting it so you could maybe do a really quick test in a room with LAMP to see, okay, is it here, yes or no? And we're working on that with Northwell now. But, the subway is part of a broader project. It's called MetaSub, which is the metagenomics, and really, it used to be called metagenomics, but now it's really metatranscriptomics, because we're looking at RNA. So now I think we have to recall the consortium or rename it as metaomics of subways and urban biomes, is what it stands for, MetaSub. And then it links to, within that group, there's MetaMed, so looking at medical environments, MetaSu, which is sewage, MetaCats, looking at how many cats are carrying coronavirus. We're launching that this week. So if people want to know what's in their cats, I think so. I'm curious. We have 2 cats. I've looked at them suspiciously ever since we knew that cats can carry it. Although, to be clear, there's no evidence of feline to human transmission yet. So I don't. It's usually the other way around. Usually we're just spewing it onto our cats, apparently. But there's very limited data. So anyway, yeah, we've done a little bit of subways. That's in review as well, and more work coming from the hospitals. We use with the same protocols.
Do you think the subway study will go global as?
It is. It'll go viral. So it has already. Actually, we have now 12 different hospitals repeating the MetaMed study, and 50. It should be 51 cities that'll repeat the surface sampling. We just got some of the first plates back from Krakow in Poland, and we're getting some from the UK tomorrow. And so we're going to do a uniform extraction and processing. Actually, all those also with the NEB-NIX ribodepletion RNA-seq kit. So we're doing LAMP on them, basically aliquoting and doing LAMP and then RNA-seq. And we just got the first 300 samples back, actually. That was a lot of like using the low input hospital samples. And the only challenge with low input RNA is you have to do a little bit more PCR because there's not that much material. So we've been doing 18 cycles. So it increases the duplication rate when you do sequencing. And it's a bit technical. I don't know who's going to be in the audience for this podcast, but as a caveat, that's when you go to low amounts of RNA, the only challenge is you have to amplify more, and then you get a little bit more duplication rate. But I just got a link last night from David Denka in the lab who's working on the data, and you can very clearly see, though, the RNA-seq picking up the SARS-CoV-2 from the hospital sample. So we'll get more from the global. We'll probably finish all the extraction, in the next few weeks, and then sequencing over the summer. But it'll be a really big global data set of what's on the surfaces during a kind of a once-in-a-century pandemic.
Wow. Are you finding it in places that are unexpected? Even in hospitals, like, wow, I didn't think it would be there.
Yeah, actually, interesting. I maybe at some point I have to. well, I could give a talk with slides to show it. But interestingly, one of the more surprising places was, we see it, like, for example, the bedpans in the patient rooms. And we actually just got the updated draft of this paper. So again, this will probably be at least in a preprint for, depending on when this comes live, we could send a link to it. But we can see it, next to the bedpan. It's in when the patients go to the bathroom. It's in their bathroom when they're COVID positive. It looks like it seems to be somewhat correlated with the CT threshold that was found in the patient, but not always. Sometimes people are super spreaders but don't have as much, it seems, in their NP swab. But the surprising thing was really that in the bathroom, you'd think, where would it be? It's definitely there, but the place that had it the most was actually on the ceiling. And we think it's because most of the toilets in hospitals are openers. So when you flush it, you basically just aerosolize whatever's in the stool. And I think it goes to the ceiling. So we actually swabbed this. And so, yeah, you're making an appropriately gross face. That's I don't know what to call it, except for like nuclear fallout can happen. It's like fecal fallout is what I think this is. Yeah, so that was surprising, and that's we're looking more into that. But we're sequencing those samples as well as the RT-PCR.
Yeah, that's disgusting.
So, there's this big debate about should men close the toilet lid down or not, and men don't do it, or who should do it. Like, there's no ambiguity here. Everyone should just close the toilet. Like, there's anyone who doesn't do it is failing humanity. So just everyone put the lid down, please. Yeah.
I couldn't agree more. So you mentioned briefly that you're involved in some COVID research consortiums. You've got together some. I know you have a lot of collaborations. So you mentioned GLAMP, or is it glamping?
It's the GLAMP consortium, I suppose. Yeah, and some meta subs.
Could you tell us a little bit more about those groups that you're involved with?
Yes, so both. The GLAMP consortium is a public-private partnership, if you will. It is anyone's welcome. Actually, explicitly defined it as a pre-competitive space for people to share ideas. So people may take some of those ideas and commercialize them or optimize them, tweak them. That's great. It's similar to what actually NIST does this for some standards groups, the National Institute of Standards and Technology. They often define their work as a pre-competitive space. So get all the industry players together, the government groups, the academics, and say, this is a place for exchange of ideas that is before you go out to try and commercialize something. So, and that is the ethos and sort of the structure behind the GLAMP group. It's open to anyone. It's just a call to share ideas. There's a Slack channel, a Google group, and an exchange, and it really is meant to be an open forum for thinking about ways to address the pandemic. So that's been. Again, it just started because I kept emailing people, and so I just took everyone I was emailing with and made one big list and said, okay, let's all talk once a week. But also, we've been chatting a lot with Eileen and other NEB scientists, also very frequently, sometimes daily, depending on protocol iterations, to think about how do we get saliva to work. And it is, it's been a really great collaboration within NEB in our lab, and then also with the whole consortium. And there's one other group called Testing for America, which is a nonprofit I helped found a month ago during this sort of pandemic to help also invest in more testing methods. So that's a philanthropic effort just to get more investment in, variations of LAMP, for example, or another protocol called LAMP-seq, where you actually can do a quick LAMP reaction with a barcode, or a swab-seq, or even nanopore methods we've been looking at just came out, which we had some data about 3 weeks ago, and then Oxford Nanopore had a LAMPore protocol that just got released. So, but all these are, I think, in the early stages of this pandemic, we don't know what's going to be the best protocol, what's going to be the most scalable. So let's launch as many ships as possible and support them all at launch. And then we don't know which ones will make it the farthest in the ocean of uncertainty, I guess is where this metaphor is going. But want to have an exchange where everyone can at least launch their boats and then get as far as they can.
Absolutely. Thank you so much for, I think, your efforts in that. Certainly the coronavirus pandemic has brought to light the importance of global collaborations, I think.
Yeah.
And a lot of the individual scientists wouldn't be able to make the great leaps and bounds, I think, in discovery without the immediate sharing of information from the preprints and without these global collaborations. And thank you for setting up consortiums to help answer everybody's questions and to help, drive the research forward.
Yeah, I feel like it's my duty as a human, I think, but it's also a sanity check. It's much more efficient to have a call with 100 people interested rather than 100 individual calls. So some of it was just for my own sanity, but it's been really great to see people. It's exactly what I think when we were all kids thinking, oh, I want to be a scientist, and science is collaborative, and you have ideas, and there's no ego, and everyone's just hoping to make the world a better place. It's maybe the closest I've seen to that ideal scientist space that scientists have ever been. But there's still egos and still people trying to do the best of one thing, or get the fastest or best paper or method, or some people also trying to, a lot of people are trying to pivot their business to somehow survive in COVID, right? So people that have companies, some of them are in this because they need to do something. Whatever business model they had before may no longer be relevant, or it won't be relevant for another 6 months, right? So it's a really unusual time, but it has been the most selfless I've ever seen the entire global research community. It's been really inspiring.
Yeah, some silver linings amid the crisis.
Yeah.
Nice to see. Thank you so much for taking time out of your schedule to join us today, Chris.
Great. Oh, thank you. It's a pleasure, and thanks for making so many awesome reagents and putting them onto fountains that you can grab enzymes on. I've heard rumors that there's fountains of enzymes on campus that just shoot up, and you can take a cup of and get your enzymes. Is that true?
Yes, totally. Yeah.
Okay, good. That's what I've heard. Good.
Delivered by unicorns. That too.
Great.
You'll have to come see for yourself.
Yes, yeah, I would love to come back. I've been there a few times, I guess, for the 3 times, I think, and so I'd love to come back. Once we can start to see other humans again in person, I'd love to come back. Yes.
Well, we'd love to have you. Thanks again, Chris.
Great. Thank you very much.
Hope you enjoyed this episode of the COVID-19 Researcher Spotlight series. Catch our new episode in a couple of weeks when we switch back to podcast format and I interview Dr. Nama Giva-Zatorsky of Technion, the Israeli Institute of Technology. Nama's group developed a protocol for detection of SARS-CoV-2 RNA directly from nose and throat swabs, as well as self-collected saliva samples, without the requirement for RNA purification. This method utilizes reverse transcription loop-mediated isothermal amplification, offering rapid results to enable successful community surveillance.